Hello. Now that you have reached the secondary level of knowledge as per school curriculums, you are going to get an insight into what life is all about, and in our study of biology you are going to deal with the attempts that you make for understanding what life is all about. Now when we talk about life, we have to deal with its most fundamental aspects. Now for example, if I give you simplest example of a wall is made up of bricks. So the constituent part of the wall, basically what wall is, that wall is being made by bricks, and bricks are its fundamental blocks, if we can say. And to put it in simple terms, we can call them building blocks. Now when we talk about something which is living, or I would say that when we confine our knowledge in the fields of biology, we have to deal with something which those things which are living. And when we deal with living things, we are going to deal with the most fundamental unit of life, that is cell. And when we talk about cell, we are dealing with the cytology, basically. This is the term. Cyto refers to cell, and as in the case of biology, logy refers to study. So we have cytology, that is the study of cells. Okay, keeping this aside, what we have next is, by the end of this lesson, we have to arrive at the answers of these two questions. First is that who discovered cell and how come the discovery came, and the second question is that whatever discovery was made, does it suit our statement that cell is the structural unit of life, that means the most fundamental unit of life. So we have to arrive at the answer of the question that why is cell called structural and functional unit of life. Now from this whatever is mentioned over here, one thing is clear, that certainly it is the cell which is called the structural and functional unit of life. And whenever I talk that something is unit, that means whatever activity is being pursued, it would be pursued at the most fundamental level at that particular condition, or at that particular stage, or at that particular involvement, or you can say that particular arrangement, to be very specific in this case. So we are to deal with an arrangement which would be the structural and functional unit of life. Now before we come to the second question's answer, we have to deal with the history, basically. And what you can see over here, that I have mentioned few years, and these numbers, they are basically the years that are, I didn't mention CE over here. This is century wise how the cytology evolved. Now one thing I need to bring to your notice is that cytology has taken a leap, manifold step leap, that you can say that all of a sudden there was a slow, very gradual change or gradual understanding or gradual increase in knowledge. But with the advent of electron microscope, now you must be knowing what an electron microscope is. If you don't know, you just have to keep in mind that it would magnify as much as 100x, okay, or more than that. That means you can see the most detailed, even a tip of hair can be magnified and it would appear like a huge ball in front of you if you observe it under the electron microscope. Again, I am giving you the reference example. Take the tip of the hair, put it under electron microscope, it would appear as huge as a football. I would give you the example. So such is the magnification. In 1940s, there was advent of electron microscope. Now when we talk about electron microscope, we come to the field of microscopy. Now what is microscopy? You know that it is the art, you can say, it is an art of magnifying the things. And when we deal with cells or whatever living structural unit we are talking about, it is highly small structure. It is very minute structure which needs magnification. As in the case, if you have to see something very small, you take that particular lens, magnifying glass, for observing it. So is in the case of cells. If we have to observe them, we observe it under the microscopes. Now I need not to tell you what a microscope is all about, what a microscope looks like. Everybody in your age group, at your level of studies, knows what a compound microscope is. Okay, so this compound microscope on an average gives you a 45x or 10x to 45x magnification. Now what this 10x and 45x means is that they increase the size of the image whichever is being formed. If the object is 1 mm, you are going to observe it as much as 45 mm after you observe it under the magnifying compound microscopes glass. So basically these microscopes are meant for increasing the size of the object whichever is being observed manifold. And when we talk about electron microscope, I gave you the example, it is many fold you can say. Now we are going to see how this study of cells, how the discovery of cells, how just giving the actual work that is being performed by the cells, that means it is assigning their actual specificity in the body, how it was gradually being discovered. So we have the name of first person, that is Robert Hooke in 1665. First of all observed cells. Where? In cork. Now what is cork? Those who play cricket, they must be knowing there is a cork ball, that is you often use it instead of leather ball. Otherwise you must have observed as cork screw, which is present in the bottles for closing the wine bottle specially. So we have this cork, it is nothing, it is just the bark of the tree. You can take it, the simplest thing cork is, you are going to study what cork is, what is its structural composition in your higher classes, but for now what you can keep in your mind is that hard and rough portion of the tree trunk that we call as bark, that can be referred to as cork over here. Now you must be knowing that this cork is dead. Okay, the part of the plant which is known as cork is dead. It comprises of dead cells. So Robert Hooke was the first one who observed the cells in a cork. Okay, he observed it under the magnifying glass, and what he saw over there, that there were certain blocks and he named them as cells. Okay, so the constituents which formed the cork, they were something like this, a meshwork sort of thing was there. Cork was made of these units, the structural units of the cork, honeycomb like structure you could see. This honeycomb like structure was what he observed, and he named them as cells. Okay, now these cells, they were discovered, but they were not discovered in living entity. They were not living cells which were discovered. It was in 1674 that a scientist known as Leeuwenhoek, he discovered a living cell in the pond water. Now if he had observed a living cell in pond water, it must be some plant cell or algae cell that we are going to study. He observed this. Okay. Later on, as soon as the magnifying ability increased, there was more and more development in this optics field, so that magnifying glass with higher magnification were made. They could reach to a level where the internalmost part, that is nucleus, was discovered by Robert Brown. Robert Brown was the scientist who discovered nucleus inside the cell. Now what is this nucleus? You can call it, nucleus is the main principal organelle. Now what is an organelle? We are going to get introduced to that as well, but for now it is the principal part of the cell basically. If the cell is a huge sphere, you can consider it the central part, heavy central part is the nucleus. Later on in 1839, when all these discoveries were made, there came the there came two people, there were two biologists. What was their name? One was Schleiden and other was Schwann. One of them was a botanist while the other one was a zoologist. Now one of them was studying the plant cells, another was studying the cells which were present inside the animals. These two scientists, one in 1839 and 1838, they gave the cell theory. Okay. Now what did this cell theory say? This cell theory said that the cell is the fundamental unit of life, that inside every living organism you are going to find the cells, and the main work, that is at the grassroots level, if you have to see that how the organism is working, the grassroots level would be the cell. It would be the cell at whose level the actual activity would be going on, and then there would be organization, that is structural organization, which would vary from cell to tissue to organ system or organs as we call it. in the same year, there was another term which was discovered that you need to know before we move any further. There was a scientist known as Purkinje, and he discovered, not discovered, it was long before discovered, he gave the term protoplasm. Now, whenever you hear this term in future, what you have to remember is protoplasm is cytoplasm of the cell and plasma membrane. Okay? This is what we understand it in terms when we are very much aware of what the cell organelles are. But for now, what you have to remember is protoplasm is the living part of the cell. Now, you know one thing, that there are certain parts in the cell which are non-living, and one of that is cell wall. Remember it, we are going to learn about it as well. The living part of the cell would be protoplasm. Now, supposedly this is our cell, this is the plasma membrane, this is the inner content, as I told you, as not I told you, Robert Brown told all of us that there is a central part, that is the nucleus. Supposedly this is the nucleus. Whatever lies between the plasma membrane and the nucleus, that is known as cytoplasm. Okay? And in case this plasma membrane is surrounded by a cell wall, then it would be known as protoplasm, this living part, because this plasma membrane and cytoplasm, both of them are living. And how do we define something if it is living or not? There is metabolism that is going on. So we find metabolism, that is breakdown and making up of new biomolecules, that is taking place in the plasma membrane as well as the cytoplasm. I can understand that these terms would be confusing at this point, but by the time you arrive at the end of this chapter, you will be quite aware of what all these are. But for now, what you have to remember is that in around 1839, this person, Purkinje, he gave the term protoplasm. And what is protoplasm? Protoplasm is the living part of the cell. Okay. Now, moving further, in 1855, there was another scientist, that was Rudolf Virchow. You see, there was lot of renaissance in field of biology at that time. This Rudolf Virchow, he and these two, whatever they had proposed, that eventually gave the term cell theory. Cell theory, as proposed by Sheldon and Schwann, did talk about that the fundamental unit of life was cell, but they did not tell that how this fundamental unit of life is going to perpetuate itself. That means where it came from. And this person, he came and told that whenever a new cell is going to be formed, it is going to arise from a pre-existing cell. So it was also clear that a new cell is formed from a pre-existing cell. It is the cell itself which gives rise to the new cell. So there was another thing that came into being, or came into the understanding, that cells are also the fundamental unit for reproduction of life. That means giving rise to new life would also lie in the hand of the cells itself. So we get that the living part, that is living part of the organism, is protoplasm. Protoplasm is the part of cell. So we have structural unit of life, that is cell. So you see, we are coming to the answers that the structural unit of life is cell. So we see it over here. Then we have the functional unit of life. Now what is the purpose of life, if I ask you? Purpose of life is perpetuating more and more. If we talk in the simplest way, life is nothing, it is unsuccessful if it could not give rise to new life. Supposedly, just give it a thought that whatever life form was present on earth in the beginning, if it was not able to reproduce, how could we have reached a stage where the human mind is so advanced? We would have stayed those bacterial elements and nothing would have reproduced. If the organism is not able to reproduce, it is not considered as a successful life form. So, because every cell can give rise to a new cell, as proposed by Rudolf Virchow, which was given as omnis cellulae e celluli. Now this is language which you might not have ever heard. This is the language that is Latin, and he said that a cell would give rise to a new cell. That means a cell would be the main unit for reproducing. A cell is not going to erupt or emerge on its own, it is going to be formed by a pre-existing cell. So we come to the conclusion that structure and functional unit of life is cell itself. It is the fundamental unit of life where all the life activities at the most basic level would be conducted. And who discovered the cell and how, that we have seen over here. Not just who discovered the cell, we just came to know that who discovered the dead cell, who discovered the living cell, then later on who discovered the nucleus, then what was given in the cell theory by these three people. Two of them, they just said that this person, he studied that what a plant cell is, this person studied what an animal cell would be, and he proposed that a new cell could not be formed if there is no pre-existing life. So by understanding this, I hope the two questions that we started this lesson with are quite clear to you now.